Welcome to the deep dive. Think about our world today. So much of what we do, powering homes, fueling cars, it really still leans heavily on these finite fossil fuels. It
Yeah.
feels like a constant struggle, doesn't it?
It absolutely does, and yet you just look up, and the sun is bathing our entire planet in this enormous, practically limitless supply of energy every single day.
Hmm.
We've got solar panels, sure, but honestly, they're still, well, pretty inefficient compared to what nature's been doing for billions of years.
Right, and that's exactly where we're headed today. What if we could really understand and maybe even harness the sun's energy, not just with our tech, but through that incredible ancient process that plants, algae, even some bacteria use?
Photosynthesis.
Exactly. Photosynthesis. It's
Yeah.
the very basis of, well, most life on Earth. And maybe, just maybe, it holds a blueprint for our future energy needs. Things like scalable biofuels.
That's the hope. So our mission today is a deep dive into photosynthesis and the chloroplast. And we're drawing heavily from Karp's Cell and Molecular Biology, 8th edition. We want to really unpack how these organisms pull off this fundamental alchemy.
Yeah, and see what surprising lessons there are for our own future.
Definitely.
Okay, so let's jump in. Foundational stuff first. We, as humans, we're heterotrophs, right? We rely on eating external organic compounds for our energy. It's like always needing to go grocery shopping for ingredients.
Precisely. A good analogy.
Yep.
Then you have the masters of self-sufficiency, the autotrophs.
Right.
They create their own organic nutrients from really simple inorganic molecules.
And our focus today is specifically on the photoautotrophs.
Exactly. They're the ultimate solar engineers, the ones that directly harness radiant energy from the sun.
What's really kind of mind-bending is thinking about the origins of this. Photosynthesis goes way back to an ancient Earth, long before oxygen was even a big part of our atmosphere.
Oh, absolutely. This wasn't just, a biological tweak. It fundamentally re-engineered the entire planet.
So what was it like back then?
Well, early Earth was a very different place. Life was pretty limited, kind of scraping by on scarce organic molecules produced non-biologically, very slowly. The real game changer came with the first photoautotrophs.
Okay.
They likely used hydrogen sulfide, H2S, as their electron source. So instead of oxygen, they produced sulfur as a byproduct. You can actually still find their descendants today thriving in places like sulfur springs or deep-sea vents.
Interesting. But that wasn't the oxygen-producing kind yet.
No, not yet.
Right.
The true revolution, the paradigm shift, arrived roughly 2.7 to 2.4 billion years ago with the emergence of cyanobacteria.
Ah, cyanobacteria. Okay, what was so special about them?
They figured out how to use water, H2O, as their electron source, and the waste product they generated, molecular oxygen.
For me, too. It's changed everything.
Everything. And you have to appreciate the evolutionary leap here. Pulling electrons from water is incredibly difficult chemically. It needs a really, really strong oxidizing agent. So developing that ability was monumental. It reshaped the atmosphere and, paved the way for us, for all oxygen-breathing life.
And this massive innovation, using water and making oxygen, led directly to another huge event in cell evolution, the birth of the chloroplast.
Right. The endosymbiotic theory. The idea is that an ancient O2-producing cyanobacterium was engulfed by, or took up residence inside, a primitive eukaryotic cell.
Instead of being digested, it just stayed.
It stayed. And over millions, billions of years, this symbiont evolved into the chloroplast we see today. It's pretty cool. Chloroplasts still have their own circular DNA, and they divide by fission, like bacteria. Clear relics of their past.
Although most of their proteins are now made using the host cell's nuclear DNA, right?
That's right, yeah. The integration is deep now. But the core machinery, that's the cyanobacterium's legacy. And you find these chloroplasts primarily in the mesophyll cells inside leaves. Those are the green factories.
And they really are factories. Chloroplasts are huge compared to other organelles. Karp mentions they're comparable in size to a mammalian red blood cell. And a single plant cell might have 20 to 40 of them.
Yeah, they're powerhouses. There's actually a brilliant historical experiment, a real aha moment from T. Engelmann in 1881.
Oh, yeah. What did he do?
He used Spirogyra, which is a type of alga with spiral chloroplasts, and some aerobic bacteria, a bacteria that need oxygen. He shone light on the alga, and guess where the bacteria congregated?
Right, where the oxygen was being produced, so around the chloroplasts.
Exactly. It was a direct, visible demonstration that chloroplasts were the site of oxygen production during photosynthesis. So elegant.
Okay, so let's look inside one. What's the structure?
Well, structurally, a chloroplast is quite organized. It's got an outer envelope made of two membranes.
Two membranes, okay.
Inside that envelope, you find these flattened sacs called thylakoids. They often look like stacks of coins, and those stacks are called grana.
Got it. Thylakoids and stacks called grana.
And the space inside a thylakoid sac is the lumen. Then there's the stroma, which is the fluid-filled space within the envelope but outside the thylakoids. That stroma contains the enzymes for making carbohydrates, plus the chloroplast's DNA and ribosomes.
And you mentioned something about the thylakoid membranes being unique.
Yeah, they have a very high percentage of specific glycolipids, galactose-containing ones. This makes the membranes highly fluid.
Why is fluidity so important there?
It's crucial because the large protein complexes involved in the light reactions need to move around, diffuse laterally within the membrane to interact with each other. Fluidity enables that rapid movement and interaction.
Okay, that makes sense. So let's zoom out to the big picture of photosynthetic metabolism. You mentioned earlier that the oxygen comes from water, not CO2. That was a big shift in thinking.
Huge. It was C.B. Van Niel in the 1930s. Before him, everyone just assumed O2 came from splitting CO2. But Van Niel studied sulfur bacteria, which use H2S and produce sulfur, not O2.
So he proposed a general formula.
Exactly. He proposed that photosynthesis involves splitting an electron donor H2S for sulfur bacteria, H2O for plants, and cyanobacteria, and releasing a byproduct, sulfur or oxygen. So the oxygen had to come from water.
And that was proven definitively later.
Yep, with isotope labeling experiments using heavy oxygen, 18O. They labeled the oxygen in water, H218O, and found the O2 produced was labeled. Then they labeled the oxygen in CO2, C18O2, and the O2 produced was not labeled. Case closed, a real scientific mic drop moment.
So that really reframes photosynthesis. It's essentially the reverse of what happens in our mitochondria during respiration.
Precisely. Mitochondrial respiration reduces oxygen to water, releasing energy from fuel molecules. Chloroplast photosynthesis oxidizes water back to oxygen, and that requires a significant energy input from sunlight. They're mirror image processes in many ways.
And the overall process of photosynthesis is generally split into two main stages.
That's right. First, you have the light-dependent reactions. These are the steps that directly capture sunlight energy and store it, temporarily, in the chemical bonds of ATP that sells energy currency and NADPH, which provides reducing power. These reactions are incredibly complex and, frankly, still not fully understood in every detail.
Okay, light reactions capture energy. What's the second stage?
That's the light-independent reactions, often called the Calvin cycle, or sometimes historically the dark reactions. These reactions take the ATP and NADPH generated by the light reactions and use them to synthesize carbohydrates from carbon dioxide.
But they don't only happen in the dark, right?
No, not at all. In fact, they run much, much faster in the light because several of their key enzymes are actually activated by products of the light reactions. We'll get to that.
Before we dive into the details, the scale of this is just staggering. Karp mentions that plant life converts something like 500 trillion kilograms of CO2 into carbohydrates every year.
It's astronomical. That figure, 25 times global beef production, really puts it into perspective. It's this quiet, constant, massive carbon-fixing engine that literally drives the biosphere.
Okay, let's zoom into those light-dependent reactions. How is sunlight actually captured?
Right. So light travels as discrete packets of energy called photons. The energy of a photon is inversely related to its wavelength. Shorter wavelength, like blue light, has more energy than longer wavelength, like red light.
And when a pigment molecule absorbs a photon,
An electron within that molecule gets boosted from its normal stable energy level, the ground state, to a higher unstable energy level, the excited state.
you mentioned earlier that isolated chlorophyll fluoresces, but chlorophyll in a chloroplast doesn't really. Why is that difference key?
It's absolutely key. That fluorescence from isolated chlorophyll is the excited electron basically falling back down and releasing its energy as light. In the chloroplast, that excited electron is immediately snatched away by a nearby electron acceptor molecule. So the energy isn't wasted as fluorescence, it's captured, it's harnessed to do chemical work. That transfer is the crucial first step in energy conversion.
And the main pigment doing this absorbing is chlorophyll.
Yes, chlorophyll is the star. Its structure, with that porphyrin ring and magnesium ion, and especially the system of alternating single and double bonds, The conjugated system is perfectly tuned to absorb red and blue light strongly.
Which is why leads look green to us, right?
Yeah.
It reflects the green light it doesn't absorb.
Exactly. But plants aren't relying solely on chlorophyll. They also have accessory pigments, like the carotenoids. These are often orange or red, think carrots.
What do? Are they just backup absorbers?
They do act as secondary light collectors, yeah, absorbing wavelengths that chlorophyll misses, especially in the blue-green range, and passing that energy onto chlorophyll. But they also have a really critical protective role.
Protected how?
If there's too much light energy, more than the photosynthetic apparatus can handle, carotenoids can actually take that excess energy and dissipate it harmlessly as heat. This prevents the formation of highly reactive singlet oxygen, which could otherwise damage cellular components. So they're both collectors and safety valves.
So the light absorbed by all these different pigments contributes to photosynthesis. This relates to the difference between an absorption spectrum and an action spectrum,
Oh,
right?
precisely. An absorption spectrum just shows you which wavelengths a particular pigment absorbs. But an action spectrum measures the actual rate of photosynthesis at different wavelengths. When you compare the action spectrum for photosynthesis to the absorption spectra of chlorophylls and carotenoids, you see the action spectrum is broader. It shows that energy absorbed by all these pigments is contributing to the overall process.
Okay, so lots of pigments are involved. How do they work together? You mentioned teamwork earlier.
Right. This came out of some clever experiments by Emerson and Arnold back in the day. They flashed light on chlorella algae and measured oxygen output. They found something surprising. Only about one O2 molecule was produced for every 2,500 chlorophyll molecules present, and it took about 8 photons per O2.
2,500 chlorophylls for one O2. That seems inefficient if each one worked alone.
Exactly. It strongly suggested that the chlorophyll molecules weren't acting independently. Instead, they function together in photosynthetic units. The idea is the antenna system.
Like a satellite dish?
Sort of, yeah.
Yeah.
You have hundreds of chlorophyll and carotenoid molecules acting as a light-harvesting antenna. They absorb photons, but they don't do the primary chemistry themselves. Instead, they rapidly transfer the excitation energy, not the electron itself, but the energy from one pigment molecule to the next.
How does it get directed?
There's an energy gradient. Energy can only be efficiently transferred to a pigment that absorbs light of equal or longer wavelength, which means lower energy. So the energy naturally funnels downhill toward the special pair of chlorophyll molecules at the heart of the unit, called the reaction center chlorophyll.
And that's where the actual electron transfer happens.
That's where the magic happens. The reaction center chlorophyll is where the light energy finally gets converted into chemical energy by transferring an excited electron to the primary electron acceptor.
Now, you mentioned there are two photosystems involved working together, PSII and PSI. Why two?
It comes down to the energy levels. To pull an electron from water, which holds onto its electrons very tightly, it's got a very positive redox potential, and boost it all the way up to the energy level needed to reduce NADP+, which has a very negative redox potential, requires a pretty big energy jump over 2 volts, electrically speaking.
And one photon doesn't have quite enough energy for that whole jump.
Right. A single photon of red light, say around 680 nanometers, only provides about 1.8 volts worth of energy boost. It's not quite enough to bridge that entire gap efficiently. So nature evolved a two-stage system.
Like a two-stage rocket for electrons.
That's a great analogy. Photosystem 2, PSII, whose reaction center chlorophyll is called P680 because it best absorbs light at 680 millimeters, does the first boost. It takes electrons ultimately derived from water and lifts them to an intermediate energy level.
Okay, stage one. Then what?
Then photosystem I, PSI, with its reaction center P700, absorbs best at 700 millimeter, takes those electrons from the intermediate level and gives them the second powerful boost, lifting them high enough in energy to reduce NADP+ to NADPH.
And this overall pathway with the two boosts looks like a kind of sideways Z on an energy diagram.
Exactly, hence the name Z scheme. It describes the overall flow of electrons from water up to NADPH, powered by those two light-driven steps in PSII and PSI.
And you emphasize that the core light reaction is that initial electron transfer.
Yes. That moment when the excited reaction center chlorophyll, P680 or P700, passes its high-energy electron to the primary electron acceptor, creating a positive charge on the chlorophyll and a negative charge on the acceptor. That charge separation is the fundamental conversion of light energy into chemical energy.
Okay, let's dig into PSII a bit more.
Yeah.
You said it pulls electrons from water.
That sounds tough.
It is tough.
Yeah.
Splitting water, photolysis, is thermodynamically very difficult. In a lab, you need high temperatures or strong electrical currents. Plants do it using light energy at room temperature. It's remarkable.
How does it work?
So light excites P680 in PSII, making P680. This P680 loses its electron almost instantly to the first acceptor, pheophytin. Now you have a positively charged P680 plus TOF, which is an incredibly strong oxidizing agent, strong enough to rip electrons away from water.
And where does that water splitting actually happen?
It happens at a catalytic center associated with PSII called the oxygen evolving complex. This complex contains a cluster of manganese, Men, and calcium ions. It's linked to P680+ via a tyrosine amino acid residue called TRZ.
The current model, the S-state hypothesis, suggests this M and K cluster acts like a capacitor. It accumulates 4 oxidizing equivalents, one by one, as PSII absorbs 4 successive photons and P680+ pulls 4 electrons via TRZ. Once it's accumulated those 4 charges, it catalyzes the simultaneous oxidation of 2 water molecules, releasing 4 electrons, which go to replenish P680+, 4 protons, and 1 molecule of O2.
Wow. So it builds up charge over 4 photon hits and then boom, splits water.
That's the idea, and you can actually see evidence for this in experiments where you flashlight the O2 production indeed peaks on every 4th flash.
And the protons released from splitting water go where?
They're released into the thylakoid lumen, the space inside the thylakoid sac. This is crucial because it starts building up the proton gradient that will eventually drive ATP synthesis.
Got it. So PSII splits water, releases O2, generates protons in the lumen, and sends electrons on their way. What happens to those electrons next? How do they get to PSI?
The electrons pass from pheophytin through a couple of plastically known molecules, PQA and PQB, right within the PSII complex. The second plastically known PQB picks up 2 electrons and also 2 protons from the stroma, the space outside the thylakoids, to become fully reduced plastoquinol, PQH2.
Ah, so that step consumes protons from the stroma, also
Yeah.
contributing to the gradient.
Yes, exactly. This PQH2 is then released from PSII and acts as a mobile carrier, diffusing through the thylakoid membrane until it bumps into the next complex in the chain, the cytochrome B6F complex.
Okay, what does cytochrome B6F do?
Cytochrome B6F takes the electrons from PQH2. As it oxidizes PQH2 back to PQ, it releases the protons that PQH2 picked up into the thylakoid lumen. And importantly, similar to the Q cycle in mitochondria, cytochrome B6F also actively pumps additional protons from the stroma into the lumen, so it's a major contributor to that proton gradient.
Double proton contribution there. Where do the electrons go after cytochrome B6F?
They're passed to another mobile carrier, a small, water-soluble, copper-containing protein called plastocyanin, which floats around in the thylakoid lumen. Plastocyanin then carries the electrons over to PSI.
Delivering them to P700 to replace the electrons PSI loses.
Precisely. Now we're at PSI operations. Light excites P700 to P700. This high-energy electron is passed through another series of acceptors within PSI. First, A0, thought to be a special chlorophyll, then pheloquinone, A1, and then through 3 iron-sulfur clusters.
Lots of steps. Where do they end up?
The electrons are finally transferred to ferredoxin, another small mobile iron-sulfur protein, this time located on the stromal side of the membrane.
And ferredoxin is what reduces NADP+.
Almost. Ferredoxin carries the electrons to the enzyme ferredoxin NADP reductase, FNR. This enzyme takes 2 electrons, delivered one at a time by 2 ferredoxin molecules, and uses them, along with a proton taken from the stroma, to reduce NADP+ to NADPH.
So that step also consumes a proton from the stroma.
Right.
And they're really building that gradient from multiple angles.
They really are. And it's worth noting, ferredoxin is a bit of a hub. While its main job here is making NADPH, it can also pass electrons to other pathways, like reducing nitrate or sulfate for assimilation into amino acids.
Okay, let's quickly recap the light reactions in that proton gradient. The overall net reaction is something like 2 water molecules plus 2 NADP plus light energy gives you 1 oxygen molecule plus 2 NADPH. And then the H plus goes to the NADPH. So 2H2O plus 2 NADP plus 8 photons, 82 plus 2 NADPH plus 2H plus little. And crucially, that proton gradient across the thylakoid membrane gets built up in 3 main ways. 1, protons released into the lumen from water splitting by PSII. 2, protons pumped from the stroma to the lumen by cytochrome B6F as it oxidizes PQH2. And 3, protons consumed from the stroma during the formation of NADPH by FNR and the reduction of PQ by PSII.
It's intricate. And you mentioned this chain as a target for herbicides?
Yeah, some common ones work by blocking electron flow. Diuron and atrazine, for example, block the plastoquinone binding site on PSII. Paraquat accepts electrons from psi, but then reacts with oxygen to produce highly destructive reactive oxygen species. It shows how vital uninterrupted electron flow is.
Okay, so we've got NADPH made, and we've built up this massive proton gradient across the thylakoid membrane. How does that gradient get used to make ATP?
Through photophosphorylation using an enzyme complex called ATP synthase. The chloroplast ATP synthase, often called CF1-CFO, is remarkably similar in structure and function to the one found in mitochondria.
So it spans the thylakoid membrane.
Yes. It has a base part, CFO, embedding the thylakoid membrane, forming a channel for protons, and a head part, CF1, that sticks out into the stroma. Protons flow downhill energetically from the high concentration in the lumen through the CFO channel and out into the stroma.
And that flow powers ATP synthesis.
Exactly. The proton flow causes part of the enzyme, a central stalk, to rotate. This rotation drives conformational changes in the CF1 head, which catalyzes the synthesis of ATP from ADP and inorganic phosphate in the stroma.
You mentioned something interesting earlier about the proton motive force in chloroplasts being mostly pH gradient.
Right. In mitochondria, the proton motive force driving ATP synthesis is a combination of both a pH gradient, difference in H+ concentration, and an electrical potential across the inner membrane. But in chloroplasts, it seems the counter movement of other ions across a thylakoid membrane largely cancels out the electrical potential. So the driving force is almost entirely the steep pH gradient, maybe 1,000 to 2,000-fold difference in proton concentration, representing over 3 pH units difference between the lumen acidic and the stroma alkaline during illumination.
Wow, that's a huge pH difference. Now, is there only one way this ATP synthesis happens? I think I read about non-cyclic versus cyclic.
Good point. The main pathway we've discussed, the Z scheme where electrons flow linearly from water all the way to NADP plus well, is coupled to non-cyclic photophosphorylation because it produces both ATP and NADPH.
Okay, non-cyclic makes ADP and NADPH. What's cyclic?
Cyclic photophosphorylation was discovered by Daniel Arnon. It's a sort of short-circuit pathway that involves only psi. Instead of passing electrons from ferredoxin to NADP plus 1 in psi, can, under certain conditions, pass them back to the cytochrome B6F complex.
Back into the chain.
Yeah. From cytochrome B6, the electrons flow back through plastocyan into P700 in psi, completing a cycle. PSII and water splitting are not involved.
So no oxygen is produced and no NADPH is made.
Correct. But what does happen is that as electrons cycle through cytochrome B6F, protons are still pumped from the stroma into the lumen. This proton pumping drives ATP synthesis via ATP synthase. So cyclic photophosphorylation generates additional ATP without generating NADPH.
Why would the cell need extra ATP?
Well, the Calvin cycle, which uses the ATP and NADPH to make sugars, actually requires more ATP than NADPH, a ratio of 3 ATP to 2 NADPH per CO2 fixed. Non-cyclic flow produces ATP and NADPH in roughly equal amounts. So cyclic flow likely provides that extra ATP needed to balance the books for carbohydrate synthesis and other chloroplast activities. It adds flexibility.
Clever. Okay, so the light reactions have done their job, generated ATP and NADPH using light energy. Now, how are these used to actually build carbohydrates? Let's talk about the light-independent reactions, the Calvin cycle.
Right. This takes place in the stroma. The groundwork here was laid by Melvin Calvin, Andrew Benson, and James Bassam in a remarkable series of experiments after World War II. They used radioactive carbon-14 as a tracer.
How do they use it?
They fed 14CO2 to green algae, chlorella, for very short periods, then quickly killed the cells and separated out all the radioactive compounds using 2D paper chromatography. By looking at which compound got labeled first, they could map the path of carbon fixation.
And what did they find?
The very first stable radioactive compound they could detect after just a few seconds of exposure was a 3-carbon molecule called 3-phosphoglycerate, or PGA. This is why plants using this main pathway are called C3 plants.
So PGA was the first product, but what was the CO2 actually added to?
But took a bit longer to figure out. It turned out the initial CO2 acceptor wasn't a 2-carbon molecule as first thought, but rather a 5-carbon sugar phosphate called ribulose 1,4,5 bisphosphate, ROBP.
So CO2 plus a 5-carbon molecule makes a 6-carbon molecule.
Exactly. CO2 condenses with ROBP to form a highly unstable 6-carbon intermediate, which immediately splits in half to yield 2 molecules of the 3-carbon PGA.
Ah, okay. And the enzyme that does this magical CO2 addition?
That's the famous ribulose bisphosphate carboxylase oxygenase. It's arguably the most important enzyme on Earth. catalyzing the first step of carbon fixation.
But you mentioned earlier it's surprisingly inefficient.
Shockingly inefficient, actually. It has one of the slowest turnover numbers known for any enzyme. It only manages to fix about 3 molecules of CO2 per second per enzyme molecule. Most enzymes are orders of magnitude faster.
So how on earth does life get by with such a slow enzyme at such a critical step?
By making absolutely massive amounts of it. Plants compensate for Rubisco's sluggishness by producing tons of it. It can make up something like 50% of the total soluble protein in a leaf. It's thought to be the single most abundant protein on the entire planet.
Wow. Billions of years of evolution, and this is the key enzyme. Fascinating. Okay, so Rubisco fixes CO2 onto RuBP, making PGA.
Yeah.
What happens next in the Calvin cycle?
Well, it's a cycle because RuBP needs to be regenerated. There are basically 3 phases. Phase 1 is carboxylation, which we just discussed. CO2 plus RuBP, SE2-PGA, catalyzed by Rubisco.
Okay, phase 2.
Phase 2 is reduction. Here, the PGA molecules are reduced to form glyceraldehyde 3-phosphate, GAP. This requires energy input. ATP is used to phosphorylate PGA, and then NADPH is used to reduce the resulting molecule to GAP. These steps are essentially the reverse of corresponding steps in glycolysis.
So using the ATP and NADPH from the light reactions, makes sense, and phase 3.
Phase 3 is regeneration. For every 6 molecules of GAP produced, only 1 molecule represents net carbohydrate gain, equivalent to 1 CO2 fixed. The other 5 molecules of GAP go through a complex series of rearrangements, shuffling carbons around, to regenerate the original 3 molecules of the 5-carbon RoBP acceptor that you started with. This regeneration phase also requires ATP.
So it's quite energy intensive overall.
Very. To fix just 6 molecules of CO2 into 1 net 6-carbon sugar, like glucose or fructose, though GAP is the actual product, and regenerate the RuBP, the cycle consumes 12 molecules of NADPH and a whopping 18 molecules of ATP. That reflects the energy needed to reduce highly oxidized CO2 into a carbohydrate.
Okay, so the net output is GAP, glyceraldehyde 3-phosphate. What happens to that?
It has 2 main fates. Some GAP is exported from the chloroplast into the cytosol. There, it's usually converted into sucrose, table sugar, which is the main form of sugar transported throughout the plant to feed non-photosynthetic tissues like roots or fruits. It's analogous to how animals transport glucose in the blood.
And the other fate.
The rest of the GAP stays within the chloroplast and is used to synthesize starch. Starch is stored as large granules right there in the stroma. This serves as an energy reserve for the plant, providing sugars during the night when the light reactions aren't running, similar to glycogen storage in animals.
Now, you mentioned earlier that the Calvin cycle enzymes are light-regulated, making dark reactions a bit of a misnomer. How does that regulation work?
It's quite elegant, actually. Several key enzymes in the Calvin cycle, including the ones involved in GAP production and RuBP regeneration, are only active when the lights are on. This prevents the cycle from wastefully consuming ATP and NADPH in the dark.
How are they switched on by light?
It involves thioredoxin, a small regulatory protein. In the light, ferredoxin, which we saw gets electrons from PSI, becomes reduced. This reduced ferredoxin then transfers electrons to thioredoxin, reducing it. Reduced thioredoxin, in turn, reduces specific disulfide bridges, SS bonds, into sulfhydryl groups, SH groups, on target Calvin cycle enzymes, and this conformational change activates them.
So light-reduced ferredoxin, reduced thioredoxin active enzymes.
Exactly. And in the dark, the process reverses. Electron flow stops, ferredoxin and thioredoxin become oxidized, and the enzymes revert to their inactive state, with the disulfide bridges reformed. There's also a pH effect. The enzymes tend to be more active at the slightly alkaline pH found in the stroma during illumination because protons are being pumped out of it.
Very clever control system. But there's a dark side to Rubisco, isn't there? This issue of photorespiration.
Yes, Rubisco's unfortunate dual personality. The O in Rubisco stands for oxygenase. Besides adding CO2 to RuBP, carboxylation, it can also mistakenly add O2 to RuBP, oxygenation.
And that's bad.
It's counterproductive, yeah. When Rubisco adds O2 to RuBP, it produces 1 molecule of PGA, which is useful, but also 1 molecule of a 2-carbon compound called 2-phosphoglycolate. This phosphoglycolate is basically useless for the Calvin cycle, and getting rid of it is costly.
How does the plant deal with it?
It enters a salvage pathway called a C2 cycle, which is quite convoluted. It involves steps in the chloroplast, the peroxisome, and the mitochondrion. Along the way, O2 is consumed, and eventually some of the carbon that was initially fixed is released again as CO2. That's why it's called photorespiration. It uses light, Indirectly, as it requires ρBP, consumes O2 and releases CO2.
So it basically undoes some of the work of photosynthesis.
Exactly, it's wasteful. Under certain conditions, especially when it's hot and dry, photorespiration can cause C3 plants to lose up to 50% of their newly fixed carbon.
Why does it happen more when it's hot and dry?
Because under those conditions, plants tend to close the pores on their leaves, the stomata, to conserve water.
Okay, that makes sense. But how does closing stomata affect photorespiration?
Closing stomata prevents CO2 from entering the loop and traps the other two produced by water splitting inside. So the internal CO2 concentration drops while the O2 concentration rises.
Ah, and Rubisco isn't perfectly specific for CO2.
Exactly. Rubisco evolved billions of years ago when atmospheric O2 levels were very low and CO2 was high, so it never developed perfect specificity. It has only a modest preference for CO2 over O2. When the O2-CO2 ratio inside the leaf gets high, Rubisco starts binding O2 much more frequently, kicking off that wasteful photorespiration pathway. It seems to be an unavoidable consequence of its evolutionary history.
Interestingly, this means rising atmospheric CO2 levels today might actually reduce photorespiration and potentially boost yields for C3 crops, right?
That's one of the potential side effects, yes, though the overall impacts of climate change are, of course, far more complex.
Given this problem, have plants evolved ways to get around photorespiration?
They have.
Yeah.
Two main strategies have evolved, particularly in plants adapted to hot, sunny climates where photorespiration would be a major issue. These are the C4 and CAM pathways.
Okay, tell me about C4 plants. What's their trick?
C4 plants like sugarcane, corn, and sorghum were first noticed because the first radioactive products after feeding them 14CO2 weren't just the 3-carbon PGA, but mainly 4-carbon compounds like oxaloacetate and malate.
So they fix carbon differently at first?
Yes. They use a different enzyme for the initial CO2 capture in certain leaf cells. Instead of Rubisco, they use PP carboxylase. PEC. This enzyme adds CO2 to a 3-carbon molecule called phosphenol pyruvate, PEP, to form the 4-carbon oxaloacetate.
And why is using PPC better?
Two key advantages. First, PPC has a much higher affinity for bicarbonate, the form CO2 takes when dissolved in water, than Rubisco does for CO2, so it can work efficiently even when internal CO2 levels are very low, like when stomata are partially closed. Second, and crucially, PPC is not inhibited by O2. It doesn't have that oxygenase problem.
Okay, so PPC fixes CO2 efficiently even when O2 is high and CO2 is low. But they still need Rubisco eventually to make sugars via the Calvin cycle, right?
They do. And the way they combine PPC and Rubisco is ingenious. It involves spatial separation and a special leaf anatomy called Krantz anatomy.
Krantz anatomy.
Yeah, C4 leaves have two distinct types of photosynthetic cells arranged in concentric layers around the leaf veins, outer mesophyll cells and inner bundle sheath cells.
Okay, 2 cell types. How does that help?
In the mesophyll cells, outer layer, PEPC does the initial CO2 fixation, forming 4 carbon acids like malate. This malate is then transported into the adjacent bundle sheath cells, inner layer.
So the 4-carbon compound moves inward.
Right. Inside the bundle sheath cells, the mal is decarboxylated, the CO2 is released again. This pumps CO2 into the bundle sheath cells, creating an incredibly high local concentration of CO2 right where Rubisco and the Calvin cycle enzymes are located, maybe 100 times higher than in the mesophyll.
Ah, so they use PPC as a CO2 pump to concentrate CO2 around Rubisco, basically swamping out the oxygenase reaction.
Exactly. It effectively eliminates photorespiration in the bundle sheath cells, allowing C4 plants to photosynthesize very efficiently, even in hot, bright conditions with partially closed stomata. The pyruvate left over from decarboxylation goes back to the mesophyll cell to be recharged into PEP, which costs some ATP, but the benefit outweighs the cost in their native environments.
That's a really clever anatomical and biochemical solution, and it evolved multiple times.
Remarkably, yes. C4 metabolism has evolved independently in different plant lineages more than 45 times. It's a fantastic example of convergent evolution, showing how successful this strategy is. And as you hinted, plant scientists are actively trying to engineer C3 crops like rice with C4 features to boost productivity.
Fascinating. Okay, so C4 is spatial separation. What about CAM plants?
CAM stands for crassulacean acid metabolism, first studied in succulent plants like cacti and jade plants, which often live in deserts. They also fix CO2 initially into 4 carbon acids using PPC, similar to C4 plants, but their strategy involves temporal separation, separating the steps in time.
Temporal separation, how did that work?
CAM plants do something quite different with their stomata. They open their stomata only at night when temperatures are cooler and humidity is higher, minimizing water loss.
Okay, so they take up CO2 at night.
Yes. At night, CO2 enters the leaves, and PPC fixes it into 4 carbon organic acids, mainly malic acid, which are then stored in the cell's large central vacuole. The acidity of the leaves actually increases overnight, hence the acid metabolism name.
So they store the CO2 overnight as malic acid. What happens during the day?
During the day, the stomata close tightly to prevent water loss in the heat. Now, light energy is available for the light reactions to produce ATP and NADPH. The stored malic acid is released from the vacuole back into the cytoplasm and is decarboxylated, releasing CO2 inside the closed leaf cells.
Ah, so again, they create a high internal CO2 concentration for Rubisco, but they do it by separating initial fixation night from the Calvin cycle day.
Precisely. Rubisco then fixes this internally released CO2 via the Calvin cycle during the day under conditions of high CO2 and low O2, since water splitting inside a closed leaf will raise O2, but the concentration effect of CO2 release dominates. It's another brilliant adaptation for survival in extremely arid environments.
Amazing strategies. Okay, as we start to wrap this deep dive up, let's connect all this incredible plant biology back to the human perspective, specifically global warming and carbon sequestration.
Yeah, it's impossible to talk about photosynthesis without considering its global impact. We know the Earth's surface temperature has been steadily rising since the Industrial Revolution, with most of the warmest years on record occurring recently. And atmospheric CO2 levels have climbed dramatically, about 30% since pre-industrial times.
And CO2 is a key greenhouse gas, right?
Sure. It is. CO2, along with other gases like methane and water vapor, absorbs infrared radiation, heat, trying to escape from the Earth's surface and re-emits some of it back down, trapping heat like the glass in a greenhouse. While the greenhouse effect itself is natural and necessary for life, the enhancement of this effect due to human emissions is the concern.
And the link to human activity burning fossil fuels is clear from things like ice core data.
Very clear. Ice cores show that current greenhouse gas levels are far beyond the natural fluctuations seen over hundreds of thousands of years, correlating strongly with industrialization.
So reducing emissions is critical, but it's a huge challenge. The scale of CO2 production from power plants, transportation, it's immense.
Absolutely staggering. And solutions like electric cars still rely on how the electricity is generated. Alternatives like nuclear, solar, and wind have potential but require massive scale-up and face their own hurdles regarding cost, land use, or safety concerns. Even biofuels aren't a silver bullet, as burning most biofuels still releases CO2.
But this is where photosynthesis comes back into the picture, isn't it? As the planet's natural mechanism for carbon sequestration.
Exactly. Photosynthesis is the primary process that removes CO2 from the atmosphere and converts it into organic matter. Plants literally build themselves out of atmospheric carbon.
And it's not just the carbon stored in the plants themselves.
No, a significant amount of carbon fixed by plants gets transported to the roots, and some eventually makes its way into the soil as organic matter when roots or microbes die. And the scale is huge. Soils globally are estimated to store twice as much carbon as the entire atmosphere.
Wow. So protecting forests and soils is critical for carbon storage.
Absolutely critical. Deforestation, often driven by agriculture or development, releases massive amounts of stored carbon back into the atmosphere and reduces the planet's ongoing capacity to sequester CO2. It's a major contributor to global warming, sometimes estimated to be on par with emissions from fossil fuel combustion.
So it seems like tackling climate change needs a two-pronged approach. Reducing our emissions through technological and societal changes, and enhancing natural carbon sequestration through better land management, reforestation, and understanding plant biology.
That's the bottom line. We need engineering solutions and ecological and agricultural solutions working together, understanding the intricacies of photosynthesis, like the differences between C3 and C4 plants, isn't just academic. It could be crucial for developing crops that are more efficient carbon sinks or more resilient to changing climates.
So wrapping up this deep dive today, what does this all mean for you listening in? We've taken quite a journey from the very origins of how life first harnessed energy, through the amazing molecular machinery of the chloroplasts, the two photosystems, the elegant Calvin cycle building sugars.
And seeing how plants have evolved these incredible C4 and CAM adaptations to thrive in challenging environments, overcoming limitations like photorespiration.
It really underscores just how fundamental photosynthesis is, not just for the plants, obviously, but for maintaining our atmosphere, regulating climate, and basically supporting almost all life on Earth.
It's the quiet, constant, planet-sustaining process happening all around us.
So here's a final thought to leave you with. Given how absolutely critical photosynthesis is for balancing atmospheric carbon, and knowing the challenges of photorespiration and the cleverness of C4 evolution, what role do you think advancements in plant science, like those efforts to engineer C3 crops to be more C4-like, might realistically play in our broader efforts to combat global warming?
It's a complex question, linking fundamental biology directly to global challenges. The sheer complexity of a single process like photosynthesis just highlights the incredible interconnectedness of life and the environment. There's so much more to explore.
Indeed. Well, we've covered a lot of ground from Karp's chapter today, hitting all the key concepts, processes, and their significance. We hope this deep dive has been helpful.
Thanks for joining us today, and thank you especially for being part of our last-minute lecture family. We hope to catch you on the next one.